When you look at this diagram of arteries and veins, try to think about the functional differences between the two. Remember that arteries carry blood away from the heart. Generally speaking, arterial blood is traveling under the high pressure generated by the contraction of the heart. On the other hand, veins are returning blood to the heart. Venous pressure is typically much lower than arterial pressure. These illustrations show that the wall of the artery is much thicker in order to accommodate the higher pressure in arteries. Both arteries and veins have three basic layers in common: the tunica interna, tunica media, and tunica externa. The difference in the thickness of the walls is mostly the result of a thicker tunica media layer. The tunica media is composed primarily of smooth muscle, but also includes two layers of elastic connective tissue. The two elastic lamina of the artery can be seen in the photomicrograph cross-section. The dark staining zigzag lines of the elastic lamina stand out prominently, with the smooth muscle in between. One other feature to point out in the illustration of the vein is the presence of valves in the vein. We typically find several of these cup-shaped extensions of the tunica interna in veins occurring in the limbs. Since gravity tends to work against the flow of blood toward the heart, and the blood is under low pressure, these valves help to prevent the backward flow of blood as it returns to the heart. Varicose veins, occurring in veins in the lower portion of the body, happen for several reasons. All of these reasons relate to the fact that blood must return to the heart against the force of gravity. The thin-walled veins sometimes fail under pressure, causing blood to accumulate, which enlarges the veins beyond normal size. Valves that fail to prevent backflow increase the problem. This diagram illustrates the factors which determine the bulk flow movement of fluids through a capillary wall. To understand capillary exchange, it is important to have a basic understanding of the forces involved in moving water, the predominant component of blood, from one compartment to another. Capillary filtration is the movement of fluids out of the capillary, while reabsorption is the return of fluids into the vessel. There are essentially two forces involved: hydrostatic pressure, which is derived from the pressure of the fluid itself, and osmotic pressure, which is dependent on the solute concentration of a solution. At the arterial end of the capillary, the blood hydrostatic pressure is greater than blood osmotic pressure, and this imbalance acts to push or filter fluid out of the capillary. Although the hydrostatic pressure of the interstitial fluid outside the capillary does provide an opposing force to the blood hydrostatic pressure, it is relatively small, and the result is a net filtration pressure that favors pushing fluid out of the capillary. As the blood flows through the capillary, it passes over a longer length of vessel surface and therefore experiences greater resistance. Greater resistance causes the blood hydrostatic pressure to decline at the venous end of the capillary. On the other hand, the blood osmotic pressure is unchanged at the venous end. This results in an imbalance of forces favoring blood colloid osmotic pressure, which pulls or reabsorbs water back into the capillary. The reabsorbed fluid at the venous end almost equals the amount filtered at the arterial end, and the net loss of fluid in capillaries is quite small. This flow diagram shows the two mechanisms that can raise blood pressure. These mechanisms are increased cardiac output and increased systemic vascular resistance. The factors shown on the left side of the diagram affect cardiac output. They can be divided into two groups, since cardiac output is dependent on heart rate and stroke volume. Factors that increase heart rate include hormones and the autonomic nervous system. Factors that increase stroke volume include the autonomic nervous system and increased venous return back to the heart. The right side of the diagram shows factors that affect systemic peripheral resistance in the blood vessels. An increase in resistance can be achieved by increasing the viscosity or thickness of the blood through the addition of blood cells. increasing the length of blood vessels, and reducing the diameter of blood vessels. Note that the alteration of blood vessel diameter is the only option that can be done rapidly. The other two options take a much longer time to achieve, but the effects last much longer. When we think about the normal course of blood flow, we expect blood to flow from the heart via arteries to capillaries, then to veins, and finally back to the heart. However, portal flow usually varies from that pattern. In a portal system, blood in veins flows into a second set of capillary-like vessels before returning into another set of veins known as portal veins, before returning to the heart. Let's take a closer look at how that works in the hepatic portal system and think about its advantages. Arterial blood is sent to the intestine to supply it with oxygen. The intestine's primary job is to absorb nutrients into the capillaries. The blood in those capillaries is now nutrient-rich but oxygen-poor. That blood is then directed toward the liver via the mesenteric veins. Blood in several other major veins, including the splenic and gastric veins, also flows toward the liver. These veins converge at the hepatic portal vein, supplying the liver. Within the liver, microscopic capillary-like vessels called sinusoids deliver the nutrients to the cells of the liver where they are stored and metabolized. The blood then returns to the general circulation by way of the hepatic vein. Because of the hepatic portal system, nutrients absorbed from the digestive system are much more rapidly transported to the liver than if the nutrients had to travel throughout the entire systemic circulation. Understanding fetal circulation requires that you have a very clear understanding of adult circulation. With that grounding, let's take a look at the differences that occur between fetal and adult circulation and the incredible changes that happen at birth. Fetal organs, such as lungs, intestines, and kidneys, are not yet doing the jobs they will do as adult organs. The fetus depends entirely upon the mother to take care of its basic needs for oxygen and nutrients, as well as eliminating wastes. Exchange of these substances occurs in the placenta, where maternal and fetal blood comes together close enough to make that possible. Highlighted in the yellow boxes are the 5 structures unique in fetal circulation. The umbilical vein carries oxygenated blood towards the fetus. The 2 umbilical arteries carry the oxygen-depleted, waste-laden blood back toward the placenta. Remember those red-blue conventions. Red represents oxygenated blood, not necessarily arterial blood. Varying shades of red, blue, and lavender indicate the relative amount of oxygen in the blood at any given point. 3 other structures, the ductus venosus in the liver, the foramen ovale in the heart, and the ductus arteriosus between the pulmonary artery and the aorta, are transient structures that close after birth. During the fetal stage, several organs do not receive blood, as their jobs are taken by the placenta. Functionally, the foramen ovale and ductus arteriosus allow the majority of blood to bypass the lungs. Similarly, the ductus venosus allows most of the blood to bypass the liver, delivering it to the general circulation so it can transport nutrients directly to the developing organs of the fetus.